Have you ever played the game called Telephone, where one person passes a message to another person and another person passes the message to another person? Well, our cells do that as well as they transfer information from cell to cell through a series of pathways. But what if one of the cells were one of the messages was misinterpreted or miscommunicated? Hi, good afternoon. My name is Ivan, and I'll be discussing GPCR ligand binding class C/3 metabotropic glutamate pheromone receptors. We'll be explaining the pathways, discussing the importance of these pathways, and the malfunctions and the solutions to those malfunctions. My group members consist of Alderico and Runches. So what is class C/3 GPCR? Class C/3 GPCR G protein coupled receptors are a specific group of receptors involved in cellular signaling that responds to a wide range of ligands, including neurotransmitters like glutamate and GABA, as well as calcium ions and sweet umami tasting molecules. These receptors are crucial in several physiological processes, particularly in the brain and metabolic regulation. Now, there's three types of GPCR, they are classified into group 1, group 2, and group 3. Group 1 consists of M group R1 and M group R5, whereas group 2 consists of M group R2 and M group R3, whereas group 3 consists of M group R4, M group R6, M group 7, and M group 8. Now, group 1 activates the GQ process in GQ, which means that it amplifies signal by activating the phospholipase PLC, leading to the production of second messages like IP3 and DAG, which causes calcium release from the endoplasmic reticulum and activate the protein kinase. And moving on to GQ, which is a inhibitory process, which these receptors will inhibit the adylate cyclase, reducing the level of CAMP, which in turn affects various cellular processes, including the regulation of neurotransmitter release. Now we'll be talking about the three processes that we pick, which is the metabotropic glutamate binding to the metabotropic receptors, the CASR binding to the calcium ions, and the beta compound binding to the T2RS. So let's start with the metabotropic glutamate receptors binding to the glutamate. As you can see, this is the pathway for the metabotropic glutamate receptors binding to glutamate. So initially, glutamate will bind to the metabotropic glutamate receptors, which causes the change in shape of the receptors, which allows the G protein consists of G alpha, G beta, and G gamma subunits undergo activation as GDP bounds to G alpha subunits and is replaced by GTP. This exchange activates the GA subunit, dissociates it from the G alpha and G beta and G gamma complex, allowing both to interact with downstream effectors, which will activate GI, which amplifies the signal as previously discussed. Now let's move on to the second process, which is the path of calcium ions binding to CASR. So calcium ions will bind to the CASR, causing the change in shape of the receptors, which it has a similar interaction with G protein, where the conformation chain of CASR allows it to couple with the heterotrymic G protein, which consists of G alpha, G beta, and G gamma subunits. G alpha subunits exchange GDP for GTP, which activates the G protein, and the G alpha and G gamma beta subunits dissociates and interacts with downstream effectors inside the cell. Now, these could go either GQ or GI. And the last one would be the T2RS bind to the bitter tasting compound. Now, as previously discussed, the bitter tasting compound will bind to the T2RS, which is a family of G protein coupled receptors responsible for detecting bitter compounds. Now, for this one, the conformation will be different when the bitter compound binding to the when the bitter ligand binding to the T2RS, you undergo a conformational change that activates the associated G protein, typically G-alpha glutathione. This is similar to the N-GLUT-R activation, where glutamate binds and lead to structural changes that enable G protein coupling. And this will lead to GI, which means they will not amplify the signal. And I'll pass the malfunctions to my group member, Rinches.
In this slide, I'll be talking about the malfunctions and consequences of the metabotropic glutamate receptors. So the first of all will be neurological disorders, which the dysregulation of metabotropic glutamate receptors can lead to disruption of the accessory neurotransmission. So what happens, as an example, epilepsy, is what happens is that overactivation of certain receptors in the metabotropic glutamate can lead to increased accessory signaling, and that leads to a seizure. And the second one that we'll be talking about is mood disorders, which happens because of the alterations to the metabotropic glutamate receptor signaling, and it is indicated that it is one of the pathophysiology of mood disorders. And next, I will be talking about addiction. Metabotropic glutamate receptors play a role in modulating our brain's reward pathway. So what that means is that there are, for example, substance use disorder. So altered receptors activities can influence the release of our dopamine, and it reinforces some of our addictive behaviors. For instance, some of the antagonists of the receptors may induce drug-seeking behaviors in some animals. And lastly, I'll be talking about taste sensation, which, as alluded before, these receptors, the metabotropic glutamate receptors, can allow us to perceive taste, and malfunction may lead to altered sensitivity or reduction in ability to perceive flavor, and it may make our food less flavorful, or like it will decrease or increase our ability to like sense taste like sweet, salty, and bitter. Despite all of these issues, there are certain solutions and interventions to conquer these problems, and I'll pass it to Alderico.
So solutions and interventions for the class C3 signaling pathway, one of them would be the usage of agonists. Now, agonists are substances that replicate the activity of a neurotransmitter or hormone to produce a response when it binds on the specific receptor. So, for example, patients with secondary hyperparathyroidism, they usually have low calcium concentration in their bodies. So agonists such as evocalcid and ethyl-salsitide, they can be used in place of the calcium ions and bind onto the CASR and produce a response similar to when calcium ions bind on CASR. Now, the agonists can also be used for the T2R and bitter tasting compounds. So some examples of T2R agonists are diphenhydramine, flufenamic acid, and denatonium benzoate. Now, what if the metabotropic glutamate receptors were to fail? The usage of allosteric modulators can be applied. Now, allosteric modulators are substances that bind on the orthosteric site of a receptor to change the responsive stimuli of the receptor. Now, these modulators are divided into two, which are positive and negative modulators. Positive allosteric modulators enhance receptor response, whereas negative allosteric modulators inhibit receptor response. This allows for fine-tuning of the signaling produced by the receptor. So, for example, patients with schizophrenia or anxiety disorders, they can use positive allosteric modulators to activate the metabotropic glutamate receptor and enhance the binding of glutamate into the receptor. So these are some examples of allosteric modulators for the metabotropic glutamate receptor 5. Now, lastly is environmental enrichment. Now, this points towards the whole of the pathway. In general, this solution can enhance the entire pathway. So by surrounding an individual in a stimulating environment, the individual can engage in social activities and interactions that can recover impaired signaling, enhance cognitive behavior, and trigger neural plasticity. These are the references and sources where we got our aforementioned information. And that will be all for this presentation. Thank you for listening and watching. And we hope that you've gained new knowledge about the Class C3 pathway.